Hi, let's take just a minute to talk about culturing viruses and different ways we can do that. To culture in a science lab means to grow a pure concentrated amount of that thing. So with bacteria, how do we grow bacteria? Well, we just put some cells onto a petri dish and give them some nutrients and they grow. The same thing does not work with a virus because a virus has to have a host cell. So you have to have some kind of cells growing already there in order to grow a concentrated sample or to culture viruses on them. So there's some different methods that we can use if we want to grow and use or study viruses. First, if they are bacteriophages or viruses that attack bacteria, first we grow the bacteria. That's what these three petri dishes are showing. The white stuff is bacteria growing in like a film or a carpet on the surface of the gel. They have nutrients and things underneath that they can soak up, so the bacteria will grow. Then you can add a solution with this is showing different concentrations, different solutions with the virus in it, and they'll infect the bacteria cells. The gray dots that you see, that's where the virus has started growing on the bacteria and killing off little spots. So you know that you can find a concentrated culture of the virus around each of those little spots. So this was a solution that had a lot of the virus in it. This was a solution that didn't have very many bacteriophages in it. But that only works for bacteriophages, because remember, viruses are very specific for the kind of cells they can recognize. So what if it's a virus that's for some other organism, like an animal? Well, one way to grow animal viruses is inside living animals. And that's what people did for a long time, because that's the technology that was available. However, growing and raising a whole bunch of animals takes up a lot of space. It's also very expensive. You basically have to have, a little farm or a kennel area for a whole bunch of animals. And there's also ethical concerns. How can you take this animal that you've bred and grown and taken care of, this healthy grown-up animal, and then infect it with viruses just to get samples of the viruses? So this is what people did for a while because that's all that was available. But this is really not used very much anymore. Almost not at all, because of those ethical concerns with, humane treatment of animals. A lot of viruses can infect more than one species. For example, flu viruses can infect different species of birds as well as mammals. So some viruses, if they're not very specific, they can be grown in embryonated eggs. That means a fertilized chicken egg. And the virus could be injected into different parts depending on where in the egg it can survive and grow. And this might cause changes or death of the embryo. Sometimes the virus doesn't kill the embryo. But that's a way that some viruses are reproduced for research. For a while, they used to make the flu virus this way. So you used to hear that if you had an egg allergy, that you should not get a flu shot. But flu vaccines. We used to make the flu vaccine by reproducing the viruses inside a chicken egg, and then we'd extract the virus and use pieces of that to make the vaccine. We no longer produce them that way. We have other types of cells that we use. So if you're looking at getting a flu shot or other vaccine, the egg allergy is not an issue anymore. However, that doesn't work with viruses that are really specific to certain types of cells. If it needs a certain kind of cell to grow, then we use something called a cell culture. This is a lot less expensive. What you do is you take a sample of some cells from a tissue, for example, like a lung or muscle tissue, and then you can put those in a solution to break the cells apart a little bit, and you can grow them in a petri dish. So these would be human lung cells in a petri dish, and then you can use those living cells. You can take samples, fill more petri dishes with them, and then you can grow the virus with those types of cells. This takes very little space. It's less expensive. It's pretty rapid. There are fewer ethical concerns. But in the past, there have been concerns about the source of the cells. If these are human cells, what person did these come from? And there have been instances where samples of the cells were taken without asking the person for permission of extra samples or for permission to use them in research. So there's some ethical debate about when is it okay to use a sample of a person's cells and reproduce them in petri dishes in labs all over the world. So there's still some ethical concerns with these, but that's how most virus research would be done today.